Good afternoon. I hope you all had a great lunch. It's a pleasure to be here today. I'm coming from Boston. It's my first time in Poland. I'm excited to be among you guys today. And so I'm going to tell you a little bit about how we're using single-cell RNA sequencing for cell type discovery. And so I've actually been doing single-cell RNA sequencing omics for the last 6 years. And I'll share with you some of the concepts and frameworks we've developed since. And so I think this morning you heard a little bit about different type of biology that can be inferred from single-cell RNA sequencing, right? Does this pointer work? Yeah. So from defining discrete cell types to perhaps unraveling mechanism of action. For example, if you perturb a system in vitro, you can decompose what's the mechanisms from what's just simply heterogeneity. You can map continuous cell type phenotypes. You can infer trajectory. I think Ola this morning also described how you can use this to map different cycling processes. You can infer spatial information, and importantly, in the context of human translational work, which is what I do, you can start comparing disease to patient individuals. But for today, we're going to talk about how you can use single-cell RNA sequencing to rediscover cell types. And so a lot of the work that we do is about trying to identify which cells drive disease. But if you want to eventually find these guilty cells, the first thing you need to do is kind of revisit your understanding of the human body cell composition. So the human body in the textbooks is thought to be composed of 30 trillion cells, of which 300 major cell types are supposed to be capturing most of these cells in the human body. But if you're an immunologist like myself, you will know that just specific type of immune cells like the T cells englobes hundreds of subsets. So in that context, we can ask, how do we define and classify immune cells, or just cells in general? And depending on the community you come from, we all use different modalities. Us immunologists, we love our surface markers. That's kind of how we go and isolate the cells. Some other people that do more development studies will, like ontogeny, transcription factors, morphology is obviously important, function is obviously important, but some of the problem in the field is that we tend to use only one of these modalities to infer, cell types and cell identity, when in fact it's actually all of these modalities that contribute to cell type identity. So when you work in human translational settings, a lot of the definitions we have about cell types come from mouse models. And that has a few problems when you do human work. The first one is purity. You don't know that the defined cell types you're using are actually homogeneous. Because a lot of the surface markers we are using to isolate these cells have come from mouse, we actually don't know whether these are the best surface markers to capture our cell types of interest. And also, among us, human individuals, we have different genetic background that can contribute how we respond to our environment, and thus this can impact our cell states, which is also not taken into consideration in the definition of cell types. So with all this in mind, we can ask, do the existing sets of surface markers that we use to isolate our favorite immune cells really define distinct cell types? And are there any subsets that are currently not being appreciated? And so I like to think that immunologists were ahead of their time, right? They already in the 1800s, they used single-cell approach here at staining and morphology to kind of define what this population would be. This is a myeloid cell. And then with technology evolution in the 50s, we had flow cytometry. So for those of you who don't do flow cytometry here, we're simply looking in 2 dimensions. We're sending for surface markers, and every dot is a single cell, and the higher the dot, the more it expresses a certain protein. And so using this type of analysis where you can look at 1 to 20 dimensions, we could go from 1 population to 5 populations being defined among the myeloid cells. But then the question is, when you use this type of candidate approach with predefined dimensions, you're limited in terms, your definition of cell types is limited by the dimensions that have been pre-selected. So something that may look like a sheep may not be a sheep, right? And so how can you go about revisiting our cell type classifications? And this is where single-cell transcriptomic came along as solutions to do that. And when applying single-cell RNA sequencing to this myeloid cell compartment, we went from 5 populations to 10 populations, and I'll explain to you how we went about discovering new cell types in human blood. And so I proposed using single-cell RNA sequencing to do that almost 6 years ago, and back in the days it was seen as being pretty ambitious. and risky. So I was told to kind of do a proof of principle. So I chose dendritic cells and monocytes to do my proof of principles for two reasons. First, dendritic cells are pretty rare. They represent only 1 to 2% of what circulates in your blood. So the way I was thinking about it, if I could succeed in discovering new cell types in such rare populations, when I moved forward to a more abundant population, it wouldn't be as technically challenging. And then monocytes being related to dendritic cells, we wanted to see how distinct they were. So just by show of hand, how many of you do immunology in the room? Oh, there's some people. This is great. You picked right. But for all of you who don't, I don't want to lose you in terms of the biology. And so I'm just going to take a few minutes to kind of use an army analogy. So for those of you who are immunologists, just bear with me because this is really just an analogy for our folks here that don't do immunology, so they can follow the conclusions. So one way to understand the immune system is using the army analogy. So most of the time you think about the immune system as a way of defining your cells from the intruders, but the immune system is also there to help protect you and keep you healthy. There's different components of the immune system. The groups of cells that are part of what we call the innate immune response. So these are the cells that can rapidly respond to an intruder, and those that are there to mount long-term memory that we call the adaptive immune system. So we're going to talk about the dendritic cells here and monocytes, macrophage. So the dendritic cells are a little bit like the cavalry, okay? They're a specialized unit of the army that can go and identify the intruders. They can take a little piece of the intruder. I won't say how, just use your imagination as a cavalry, and then they can bring it back to the camp where the generals are, and then you can show the generals, which will be the T cells, what the intruder looks like. So then the generals, which are part of the long-term memory, can mount a more specific attack. And so you have here a little video that hopefully will work, that shows you what they look like with their projections for sampling the environment. And here you have a picture of, this is an electron microscopy picture of these cells interacting with the T cells. And here you have a live video of the cavalry here interacting with the generals. And so the camp themselves would be like in your lymph nodes, for example, right? And the generals here, there's different types of generals, different type of T cells, but most of them will wait to get involved until most of the acute danger has passed on, and they're there to kind of help direct the attack more specifically towards the intruder. So here you have examples of T cells that were educated to kill cancer cells or to engulf pathogens. So this is for my little analogy. So we're going to talk a little bit about the cavalry. So they're very good at sensing the pathogens, at presenting to the generals the T cells. Monocytes, we didn't touch on them with the army analogy, but suffice to say that they're very good at eating things and at secreting all sorts of modulators to manipulate the microenvironment. Both of them are involved in autoimmune disease and cancers, and thus are good therapeutic targets. When we started this study almost 6 years ago, this is what we knew about what was in the blood. You had the plasmacytoid dendritic cells, which are known to produce interferons, which is very important to fighting cancer and infection. And then you had the myeloid cells. See, they have a different shape. These are good for instructing the generals that are CD8 T cells. These are cytotoxic cells. These are the ones you want activated to kill the cancer cells. And then you have the CD1C and the double negative, which are thought to be good to activate a different type of generals, the CD4 T cells, which would be involved more in autoimmune disease. And then we thought we had up to 3 different types of monocytes. What I want you to pay attention here is not so much a different type of cells, but actually the frequency of distribution, because we're going to get back to this. So I know this is mostly a computational workshop, but one thing I want to say here,
That's good. Yeah.
Oh, I thought you said push, you're on the line or something. But I was going to say I want to also highlight different sampling strategy, because it's important if you're not doing the experiment yourself, it's important that you develop a very close partnership with your experimental partner if you're not, leading the experiments. And I'm going to show you why in a few slides. Like, you need to think about sampling strategy, how do you analyze the data to inform subsequent experiments, and we're going to touch on this a little bit. So some of the questions we wanted to answer with this single-cell RNA sequencing approach included: how many subsets can be found in the blood using an unbiased approach? Do they have the expected markers that have been used for the last 20 years to study these cells? Can we identify better marker that could empower the community? Is there any unappreciated heterogeneity within major subsets? Are there previously uncharacterized cells that we don't know about? And then can we use our healthy map to start exploring disease? Okay, so what I'm going to do first is show you through a graphical abstract the overall strategy we take over and over again in the paper we published last year, because you can basically apply it to any type of tissue and problems you may be interested in. So first we sampled the tissue of interest. In this case, it was blood. And then usually immunologists, we kind of look in 2 dimensions with their 2 favorite markers and ask if a particular population that look homogeneous based on 2 markers look different or all the same, and we're asking this question using single-cell RNA sequencing approach. Then here, every color represents a unique transcriptional identity. We're going to show examples of how we cluster the cells based on similarity, and then run different analysis where we can compare clusters to each other to kind of define unique genes or markers that are specifically expressed to every clusters. We can reanalyze specific clusters to assess whether there is any unappreciated heterogeneity and so forth. Once we get this beautiful clustering map, which you see in every single cell paper, right? You have to keep in mind that a cluster is a prediction, right? There can be a lot of technical reasons why you're getting a cluster that is not biological. So you need to go and help the experimentalists validate this cluster. I'm going to show you a couple of examples. So how do we do that? First, we try to identify sets of genes that are uniquely expressed to the purple cell. So what you see in this heat map is, every column is a single cell, every row is a gene. Yellow means highly expressed. So obviously, we're looking at genes that are uniquely expressed in every cluster. Then we try to identify genes that are surface markers for which we could buy antibodies. Then we do a prospective isolation of cells from a similar tissue, and then we try to see if these 2 antibodies, orange and green, uniquely bind to a purple cell. And we look at this in 2 dimensions. And if we see double positive cells, either by flow cytometry or by staining, you're on the right path, but it doesn't mean you validated these cells, because remember, you've actually used thousands of dimensions to identify these cells in this cluster. So how do we go about validating this? We use this flow cytometry approach to single cell sort again these predicted purple cells in plates.
I got lost a little bit, because
Yeah.
it was just like this step number 3. Can you explain again? So there are testing antibodies to enrich for. So
I'll give you some examples, but
you will talk about this later. Yeah.
So I'm just giving you an overall strategy so that when I give results, it kind of makes sense. But basically, we use antibodies that are targets for genes that are surface protein that are predicted to be uniquely expressed in cells of a specific cluster.
And you want to say, okay, I validated that there's this cluster of specific one cell type because it was enriched for only for specific antibodies, and nowhere else were these antibodies also enriched.
Exactly.
And all of the cells in my cluster have the same set of antibodies.
Or like specific markers, because what do you want to do ultimately, beyond this nice clustering map? You want to be able to study these cells, like you want to hold them in your hands to be able to functionally characterize them. So how are you going to go from a T-SNE to actually holding the cells in your hand? Right? Like the beauty of single-cell RNA sequencing approach here is to empower us to identify new cell types, potentially, or new cell state. But then the biologists down the road that will have to study these cells to really define their identity need reagents to do that. Otherwise this whole process is going to be a little pointless, right?
And then when you mentioned this, that you want to identify new cell types, so as an expert in immunology, are you able to say now that scientists know the entire repertoire of human immune cells? Or
I wish, but that would be great. Then I could retire so early. We're working towards that. I'll show at the end how we're doing this at scale now as part of the Human Cell Atlas. I think we're just starting to kind of revisit all this. I think we definitely know, and don't quote me on this, I know I'm being recorded, we definitely know at least 85% of what's circulating. We're probably missing mostly rare cell types and rare state. And the challenge here is that it's not because it's rare in healthy individuals that it doesn't become abundant in disease, and then you don't even know you're supposed to be tracking it, right? And same goes if you're studying like cancer. There might be some very rare clones that are resistant to treatments, and then you don't even know they're supposed to be tracking them, if you don't know they exist. So it's kind of the same idea over and over again. So what I'm showing you here is a strategy to go from a predicted clusters to actually holding the cells in your hand. And so we use flow cytometry or cell sorting to do that, right? So we need to identify genes that are uniquely expressed that encode for a surface receptor because we don't want to lyse the cells. And then here, because you only look in 2 dimensions, like flow cytometry only looks here at 2 markers. You have to keep in mind that it's not because your cell expresses 2 protein that's as the same cells, because you actually use thousands of genes to define your clusters, right? So then to validate that this is the same cells, then you prospectively do single cell sorting of this gate here, and then you run RNA sequencing again. You go back to your map and you project the data. If they overlap, and I'm going to show you some examples, then you validated the existence of these cells. You've also identified sets of reagents like antibodies and a gating strategy to isolate these cells and to study them. And that's important because then you can really characterize these cells. You can hold them in your hands, which means you can actually, perturb them. You can study their interacting partners. You can look at them in situ, in tissue. You heard about spatial omics this morning, right? And this is what's going to allow you to really define if it's a new cell type or not. And so we do this iteratively to try to build a healthy map that we then use to compare to our disease map to identify groups of cells that are uniquely present in disease state. So that's the 4-step strategy, and I'm going to show you some examples for each. So do stop me, please, if something is not clear. So how did we go about discovering dendritic cells? I want to show you some example here because we made a lot of discoveries with less than 800 cells. So you hear a lot about these new technologies like emulsion-based approach droplets that, and they tell you need to do thousands and thousands of cells to make your discovery. Depending on your question, if it's very well defined, you can actually get to your answer with much less than 1,000 cells. So here we wanted to map anything that was dendritic cells. And so basically we defined it as not being a T cell, not being a NK cell. These are markers for lineage, not being a B cell, not being a monocyte. So we did use flow cytometry approach here, and we did use plate-based approach. What we did, I remember I told you to pay attention to the frequency distribution of the different cell types. Some of them were like 80% abundant, some of them were only 3%. If you had done a completely unbiased approach and just remove all of the non-myeloid cells, you would have mostly profiled the most abundant cell type. You would have missed all the rare cell types. So one way of enriching for rare cell types is to kind of develop this enrichment strategy where we use a handful of additional surface markers, 6 markers here. This is flow cytometry. Again, it's only 2-dimension. We drew very loose gate. So here the goal is not to kind of sort pure populations, to just kind of capture all existing cells. And then we sorted single cells from every gate on every plate. That is to control the batch effect. So every plate has a representation of every one of these gates. It's not clear, I see from your face. So this is a gate. So you tell the machine, you kind of drew, this experimentalist talking. So if you use an instrument to kind of pick single cells and move them into a plate, you have to tell the software where to take the cells from. So we manually draw, we call them these gates, or these like box here, to tell the software, okay, from this gate it will have a name, so CD141, please sort 8 wells with 1 cell each per plate, and it's going to be row B. So we manually pick the single cells, and we tell the software where to take them from. Does that make sense?
Because you have marked the places with these markers.
Yeah, so you could replace them with any of your markers of interest. The goal, again, here is not to say I want to get pure cells. The goal is to profile anything that would be a dendritic cell. So all of this could have dendritic cell potential, but we, instead of just profiling all of this, which would get you to only profile abundant cell population, we had additional markers to try to go and sample more of the likely rare population.
But that already by the choice of these markers, you're only going into the rare cell types that you know that can be selected.
So yes and no, right? Because when you add all of these gates together per plate, you're now capturing everything that's a dendritic cell. What you're doing is just making sure that you're not oversampling what would be the most abundant population. And I'm going to show you, we actually discovered, this is actually, by doing this, you actually are putting a bit more power or more weight on population that could be more rare. So we didn't pick the markers just for purity, we just kind of picked it for sampling. And we did the same thing for monocytes. Here we drew 3 boxes, and we sampled, or gate, and we sampled across the 3 gates for every plate. So see, like every plate got the three sampling strategy, the 3 gates. Okay, maybe this is a slide that will be easier to understand. And so I should say, like, so for our discovery approach, we analyzed 8 plates for this sampling strategy and 4 plates for this monocyte sampling strategy. Then we did SMART-SIG2 chemistry, which I think you heard this morning. It's full length RNA sequencing approach, which we sequence at saturation at 1 million reads per cell. And so a quick overview of how the data was processed. We filtered out any low-quality cells. We required a minimum of 3,000 genes to be uniquely expressed. And I want to stress out the number of genes here, because if you use emulsion-based approach, or your colleagues do, you'll notice that it's going to be more in the range of 1,000 transcript. And so on average, actually, most cells had over 5,000 transcript, which is a lot. We filtered out any genes that were detected in less than 0.5% of the single cells, and we log transform everything. Here, we're not necessarily interested in all genes that are expressed. We're trying to build classifiers. So to do that, we're focusing more on variable genes, right? You don't want the genes that are expressed everywhere. You want the genes that are more uniquely expressed in some cell types. So how did we do that? In this particular study, we bin the genes into 20 bins based on average expression level. Why did we do that? We want to give more weight, or actually a little bit more weight, to genes that are more lowly expressed. So some genes that can be great classifiers, like transcription factors, will tend to be not as abundant, so more lowly expressed, more likely to suffer from dropouts, right? So by doing the bin approach, that's a way of kind of giving a bit more weight to these lowly expressed transcript. And then within each bin, we estimated a z-score dispersion. We excluded genes that were lowly expressed, and then used a z-score cutoff of 1, ended up with close to 600 variable genes across the 742 single cells that passed QC. Then we wanted to define the number of principal components that most significantly capture the variation in the data. Ended up with only 5 principal components. And we went on to identify the clusters using the k-nearest neighbor graph-based clustering approach. And I'm going to show you the data now in 2 dimensions using t-SNE. So we started with the hypothesis that they would be up to, let's look at 7 cell types. And this is what we got. We got a little bit more. So any of the ones that are in black are the ones that were known. The ones that are in red are the ones that were predictively new cell types. So we discovered that one particular subset seems to split into 2 populations. We'll get back to it. This is a new population, as well as mono3 and mono4. So first, we checked whether the markers were expressed as expected, those that people have been using for decades, right? So what you see here is just violin plot. Every dot represents a single genes. They're stratified according to the clusters. The higher dot, the more it expresses a particular gene. So CD1C was indeed uniquely expressed, but if you look at other genes that are commonly used in the literature, some of them were definitely not uniquely expressed. So we went on to try to define more uniquely expressed markers. And so we're trying to find here classifiers, right? So we measured the area under the curve, and this is what we get. It's the same principle as you saw before. Every column represents a single cell. Every row is one of these variable genes. And now we have a list of more uniquely expressed markers. We also want to try to identify how many markers are needed to perfectly classify a cell. Let's say I blind you from the identity of these cells, right? How many genes do you need to perfectly predict in which cluster it goes?
In terms of their expression, or markers in terms of these anti?
So you can either do it unbiasedly based on expression, based on the best classifiers, or you can also do it based on surface markers if you want to empower downstream analysis. And we did both. And in this case, it's saturated with 3 markers. So a lot of the follow-up studies involved the selection of at least 3 positive identifiers. We also compared the prediction of the markers to the bulk analysis. So for every population that I showed, we also did bulk RNA sequencing, and then identified which genes were most expressed and discriminating across the clusters. And then we compare them to our single cell data. So here you have bulk data, and here you have single cell data. And what you can see that are illustrated using the single cell data. So what you can see here is bulk. The bulk analysis suggested that this gene was going to be the best classifier for this PDC population, and you can see that it's only expressed in half of the cells, whereas Grand-Zambi was predicted to be the best classifier at a single cell level. I mean, all of what this tells you is that single cell analysis is better at nominating uniquely expressed genes. One thing you can do with your data is also cross-reference them with different data sets. Here we cross-reference it with genome-wide association study data. So what we're trying to do is identify relevant cellular contexts where genetic susceptibility variants may contribute to the disease. And so just, this is like some tricks you can do with your data. And so here we identified a gene associated with type 2 diabetes to be more uniquely expressed in the double negative cells, whereas CARD11, which is associated with ulcerative colitis and atopic dermatitis, is more expressed in the new cell types and the PDCs.
Sorry, so then just your conclusion that it is like 3 markers to identify, this is both, so you can have some 3 genes whose expression will well classify your cell types, and surface markers, 3 different, some surface markers will be enough to identify the cell.
It's very context specific though, right? So of course,
for your particular data.
For my particular data set, of course, if you had done this for the whole blood, you'd also need to include a lot of markers to kind of exclude other cell types, right? So these are positive identifiers. And one thing we're trying to do moving forward is kind of put everything on a portal so people can more freely navigate the data. So that's something I'm encouraging people that are pushing single-cell RNA sequencing analysis. Then we can ask if there's any remaining heterogeneity, right? You can actually reanalyze a specific cluster and see if there's any unappreciated structure. And so we did that with this particular cluster. And so what you do is, you take these particular single cells and you reanalyze them. And when we did that, we observed 2 unique clusters. One of them was defined by a set of genes that are here that are more pro-inflammatory, and some of them were associated with more MHC class 2 genes. I mean, the biology itself, it doesn't really matter, except for the fact that here CD14, which was associated with this particular cluster, is a gene that is also associated with another cell type, which is monocytes. So we wonder here whether this heterogeneity could just be explained by contaminating cells, or whether these were indeed truly unique dendritic cell population. And so then we used the whole enrichment strategy I was telling you about. Okay, I'm going to show you a couple of examples of that. So we went back here and identified surface markers that were more uniquely expressed to every cluster. Then we did some flow cytometry. So again, here you're looking in 2 dimensions. So this is how we define dendritic cells. We took everything that was a CD1C dendritic cells, and then we looked. This is the gene for CD32B. We looked at CD32B expression, and we saw 2 peaks. We took the negative peak, and we looked at CD36 and CD163, so these 2 genes. And then we saw almost a unique population. So then what we did is we did single-cell RNA sequencing of this population, as well as the 10% the brightest CD32B cells. And when we did that, we kind of enriched for both clusters. It's not perfect, but we had an enrichment, and these are independent experiment. So then we went on to try to validate that these were indeed dendritic cells. What's unique about dendritic cells compared to monocytes is that they're capable of educating these T cells. They can induce their proliferation. So that's one way of testing whether you really have the dendritic cells in your hand. And so because now we have antibodies and reagents to isolate them, right, we can actually put them in a dish with T cells. What we do, we label the T cells with a dye, and then we kind of put the dendritic cells with them, and then we see if the dye dilutes. That's how we know whether T cells are expanding. And so that's what you have here. And as a control, we also included monocytes, which should not induce T cell expansion, and PDCs, which should not also expand T cell expansion. And what you see here is these are the 2 CD1C subsets we've identified. This is work that I've done with Mazanifa. And both of them could induce CD8 and CD4 T cell expansions, validating the fact that these are indeed unique dendritic cell population, and these are not resulting from a contamination from monocyte. Let's do another validation. This rare population was predicted to be new, okay? But we didn't know anything about them. So we did the same principle. We tried to identify genes that would be more uniquely expressed in these cells. And that's proven to be a bit more challenging because they present only 0.06% of what's in your blood. Lucky for us, they did express surface marker that were either much more highly expressed or more uniquely expressed, including AXL and Siglec6. So again, we did the same trick. We did flow cytometry analysis based on these two markers and across different patients, and we could see in 2 dimensions these populations present within the expected frequency. Then we did single-cell RNA sequencing of cells isolated from this gate, projected it to our initial data, and then it overlapped. Of course, like I'm making it sound like it's easy, but I mean, every time it takes about 4 or 5 months before it really starts working because you need to test different type of antibodies and clones and so forth. So now, same principle, we actually had antibodies and a gating strategy to study these cells. So we could ask questions such as, what do they look like? What's their communication capabilities? What are the direct interacting partners in situ? And importantly, like, where do they fit in the way we think about dendritic cells? So the first thing we did is, what do they look like transcriptionally? If you take just these purple cells and reanalyze them, do they all look the same, or is there more heterogeneity that's not appreciated? So when you reanalyze these cells, and these are different experiments, you actually see 2 clusters. One thing you can do in your single-cell RNA sequencing data, just want to mention it quickly, is derive gene site signatures to assess purity. In this particular case, we use the 10 most expressed genes. And so same principle, we tried to identify genes that were more uniquely expressed to every clusters. We identified CD123 and CD11C as being more uniquely expressed. So we took our AXL Siglec6 populations, further stratified it using these 2 markers by flow cytometry, did single-cell sorting out of these 2 boxes, did profiling again, and we could recapitulate the split. Same principle, again, you have a gating strategy and reagents to study these cells. And then we validated their existence across 10 additional individuals. Then the question is, can you develop more biological understanding of these cells? With the idea that RNA sequencing is just a profiling approach, you kind of need to add additional modalities to really understand what these cells are. And so it's published in the paper. I won't spend time on this, but doing a series of in vitro studies, we showed that these are new, unique, conventional dendritic cells. They have superior ability to induce T-cell proliferation. That's really important in the context of vaccination. They can secrete all sorts of cytokines more uniquely and so forth. We did infer some unique biological properties. We're following up on these cells. AXL is an important component of a receptor that can remove debris and dead cells. And these cells express 10 times more of these markers, genes, compared to other immune cells, suggesting that it could be very good at removing apoptotic debris. And they express 10 out of 13 Siglec family member genes. The Siglec stands for sialic acid immunoglobulin-like lectin. These are groups of receptors that can distinguish between self and non-self, so very important for sensing pathogens. This is where single-cell RNA sequencing was also very impactful. This ASDCs, AS for AXL Siglec6, when we looked at where they were hiding in the way people isolate dendritic cells, we realized that they were smack in the middle of how people isolate plasmacytoid dendritic cells. Okay, so here's the pink population. They only represent 6% of this population, but they're actually potentially very potent. So these populations are contaminating a known dendritic cell population. What does it mean? PDCs were classified as a dendritic cell in 1997 because of specific properties. They were shown that they could induce interleukin-12 production, they could induce T-cell expansion, they could express a set of costimulatory molecule that's actually important to induce T-cell expansion. And so what we did is we asked what happens to the PDCs if we remove these new populations we discovered by single-cell RNA sequencing. Well, first here what you see is that morphologically they look drastically different. And so it's really a contaminating population. And so we showed that PDCs do not secrete IL-12, PDCs do not express costimulatory molecules, PDCs have reduced ability to induce T-cell expansion. So thanks to single-cell RNA sequencing, we could show that it's actually this contaminating ASDC population within the PDC gate that can account for all of the dendritic cell properties that have been assigned to PDCs over the last decade. And we also proposed that actually PDCs classification should be revisited. They actually express way more B-cell-related transcript, and they remain the main cells producing interferons. Should you care about this? Yes, you should care, because actually PDCs infiltrate all sorts of different disease tissue, and there's actually different type of conflicting data that have been reported regarding these, the icon associated with infiltrates. And you're doing, all of you are doing cancer work. Actually, there's PDC vaccination strategy for melanoma with ongoing trials. And a lot of the T-cell inducing capabilities of the PDCs may be accounted for these very rare but potent cell types. And so just to kind of, show you an example of how the discovery of a rare cell types can have an important translational application. I want to, two more things. So I want to talk about uncharacterized populations. And so when you work with your experimentalists, there might always be some cell populations that are not tagged by any markers, right? Like we call this a dump gate. So we wanted to know what was in this particular population because we wanted to be able to say that we had characterized every existing population. So we did single-cell RNA sequencing of this population. And when we did RNA sequencing, we saw 6 clusters with this particular clusters expressing a lot of transcription factors that have been associated with development. And so we asked, could these cells include progenitor population? So we did some in vitro assay where we put these cells in vitro and saw what they could give rise after a number of days. And we did our readout either by flow cytometry or single-cell RNA sequencing of what was in the flask. By flow cytometry, looking at in 2 dimensions at a handful of markers, it looked like we were making conventional dendritic cells, but importantly, by single-cell RNA sequencing, we could recapitulate exactly the population that you find in the blood, both for this type of cells and this type of cells. And that's important here because the way we make dendritic cells in vitro for translational studies, like to empower vaccinations and cancer immunotherapy, we're using cells that look like monocytes and they never look like dendritic cells, right? So it's like a first demonstration done here. So we tried to see if we could enrich for these predicted progenitor population. And so same principle, we identified surface markers that were unique to this population. And to make a long story short, we iteratively tried different set of markers that were defined by our single-cell RNA sequencing data. We saw all of these populations did single-cell RNA sequencing of all these gates. Only the population here in 3 could generate in vitro all type of conventional dendritic cells. We then did single-cell RNA sequencing of this gate and could recapitulate cluster 6. And that's how we were the first to discover these conventional dendritic cell progenitors in the blood that can give rise to all type of conventional dendritic cells. And that's a pretty important discovery from a translational standpoint. Dendritic cells, they look very primitive. They express some level of growth factors that you can use to manipulate them. And what's important here is that they divide. Human dendritic cells in the blood don't divide, which means that they have limited therapeutic potential. Now, thanks to this single-cell RNA sequencing approach, you have a target that you can, isolate without going to the bone marrow and manipulate. So this shows you that they can actually divide, and they are obviously very primitive. Last few slides, just to show you how you can apply it to disease. And so as a demonstration, we use a blastic PDC neoplasm, which is a pretty rare leukemic disease, predominantly affecting male. I'm working with Andy Lane here. It usually presents with a small lesion on the skin, and within actually weeks, as it evolves to affect all organs, it's usually a fatal outcome. And so by the name, you can say like, oh, it looks like it's dendritic cell related, but actually there's been a lot of debate in the literature saying that it's more of a B cell or NK cell ontogeny. So we got blast cells from these patients, and we did single-cell RNA sequencing, and we projected the data on our data set. And so this is what you see here. Here, we're looking only in 2 dimensions, 2 sets of principal components. So the distance between the dot, every dot is a single cell, has a meaning. And what you can see is that when we project the 4 patient samples, they do overlap with the PDCs and some of these ASDCs. And so this is I'll zoom in on the heat map in a second, but what I want you to focus on is the fact that the 4 patients are here. Every column is a single cell, rows are genes, yellow is highly expressed. And so what you see here is that the 4 patients do share a lot of signatures with the PDCs and the SDCs. And now we zoom in on that cluster. And what was interesting here is that you could identify genes that were indeed expressed, but also those that were not expressed in the cancer cell types. And that gave us some hints about the biology that was defective in these patients. And we're following up on these leads successfully these days. We also identified sets of genes that were common to all patients and not expressed in healthy cell types, also identifying interesting leads to follow up. So using single-cell RNA sequencing, we went from this original classification. Yes?
I just didn't get, so these, there are the same cell types, but in the cancer cell type, other genes are expressed?
Yes.
Okay, so in the healthy, the same cell has a different expression signature than in the cancer?
Yes.
So doesn't that mean that all these methods that try to detect immune cells, for example, in cancer environment, they are wrong?
So here it's a blood neoplasm, right? So you cannot, we're not talking about like a solid tissue where you're looking at the immune cell microenvironment. This is a cancer that's actually affecting immune cell in the blood. Yeah.
So you think in the microenvironment of a cancer, these cells behave similar to the...
Well, they're
not affected by cancer themselves.
Well, they might be. I mean, that's a whole different question that I'm happy to address. So immune cells infiltrating the microenvironment are usually altered by the microenvironment itself. So the cell type may be the same, but actually the cell state will be different, right? It will be affected by what's in the microenvironment. So when you try to compare them to a healthy tissue, you'll be able to identify the type of dendritic cells that it's in there, but the transcriptional program will be altered and will be different from healthy. And this can actually help you understand the biology of the tumor you're studying.
But then can you give examples? What do you mean by cell type and cell state? So the state is defined by expression and type by these markers, for example, and or not? Or
yeah, first of all, like we should say, like cell type and state definition goes beyond just RNA sequencing, right? It's just like one modality of classification. A cell type will be very, I mean, this is a whole debate in the community, but a cell type will be groups of cells with very stable and unique identity, like, you know, a PDC will be unique, it won't change. But a cell state is like, or CD141DC will be very unique, but in the context of different environment, the core program, transcriptional program of the cells can be modulated to be like more reactive to its environment or more, you know, less reactive to its environment, and then they can kind of be distributed on the spectrum. A good example in immunology is like the T cells. The T cells, like, you know, it's a T cell because of the receptor, right? And that the T cell has different types. You have CD4 and CD8. They have like unique molecules on their surface, but within them you can have cells that are more exhausted. Well, they won't react as much to the environment. This is a cell state. And some will be very immunogenic, right? They kind of are distributed across a spectrum of cells. And you can actually define some of it actually using single-cell RNA sequencing. So we kind of revise the whole classification. And so I won't have time much to talk about this, but I just want to mention briefly, if you're not tuned in, that, you know, this was one of the first demonstrations for discovering new human cell type, and it's empowering now a larger effort, the human cell atlas. Actually, the Axel-6 we discussed were some of the poster child of this effort and the announcement in Nature last year. And just to mention it, so you know it's happening. I'm now leading a team that's kind of revisiting the whole human blood, the healthy blood, to try to rediscover cell types at state, because actually blood is actually the tissue you can most easily access in the clinic. And sorry, I'm just going to skip slides because I need to wrap up. But I just want to show that we have now generated close to 1 million cells. And so we're now doing this at scale using emulsion-based approach across healthy individuals with the same principle of cell type discovery and validation, with the hope of empowering disease studies such as, you know, the cancer cell types cancer types you're studying. And so, you cannot see my cursor. Sorry. I'm just being blinded. I just want to get to the last slide. So you asked about cell type and cell state, and I think one of the particular challenge is that, sorry, I had a lot of extra slides that were not part of the presentation, is we think a lot about classification using single-cell RNA sequencing, but the reality is that the technology is evolving and we need to kind of integrate all of these different modalities to kind of build our model. And this is actually where we're going in terms of cell type classification. And so moving forward, as you try to build your predictive model of cell types involved in driving your disease, you kind of need to start integrating all of different data sets. So with that, I'd like to acknowledge Aviv and Nir, that were my mentor for the dendritic cell study, and Raoul, who you may know for having developed the Surav tutorial package, who was my partner in crime, Moz for her teaching on all of the collaborators. And you for your attention. Thank you.
One more possibility, a quick question. I mean, we had a number.
Okay, that's so clear.
I have one.
Oh, okay.
So, you know, when you look at bacteria, there's these serotypes, right? And you vaccine, and one thing that can happen is that they switch serotype. That must be sort of a phenomenon that could happen here too, right? If you look at cancer, there may be sort of a completely different type of cell that now has a surface marker.
Yeah. I mean, also cells can be hijacked by the cancer. I mean, cancer can actually hijack some of the properties of the immune system and kind of completely transform themselves. So transcriptionally, they will look completely different.
Right.
Yeah.
But do you have any example of this where you can clearly see that this is not really the cell type I wanted to capture by using the surface marker?
In some particular case, what happens is that the surface markers are not unique enough. So what you end up capturing when you try to enrich for them is still a very heterogeneous population, and you notice that when you do some functional studies, which kind of, you get very mixed signal, which tells you that your enrichment strategy was not effective. Yeah. And so it's actually an iterative process. And so I have plenty of examples of things failing because this type of markers that I've selected didn't work. So on average, for validating a new cell type, it takes about 8 months. That involves a bit more than just single-cell RNA sequencing. We also do ATAC-seq to kind of try to understand how unique the profile is. We try to add functional studies to try to show that at single-cell level, every single cell is kind of following the same trend, right? You kind of try to be very thorough about it. Moving to disease is actually way more difficult because but it's where we're hoping to go and where a lot of you are already, because there are so many factors that are influencing the profile that it's actually hard to purely define the cells driving the disease, because they can hijack so many of the healthy profiles.
Right. Okay. Thanks.